Proenzymes. What are proenzymes? The only protein digestion enzymes are secreted in the inactive form as proenzymes or xenogens. If they are secreted in active form, they can digest cellular or extracellular proteins of organism itself. So they are activated only at the site of action. Example: pepsinogen in the pH2 forms pepsin. Now, once few molecules of pepsin are formed, rest pepsinogen is converted into pepsin by itself. This is called autocatalysis. What are isoenzymes? These are synthesized from different genes, but differ slightly in the and um, di- synthesized from different genes, but they act upon same substrate and form same product. 100 enzymes are isolated till now, which are isoenzymes. Enzymes and the number of isoenzymes. Alpha amylase that is obtained from wheat endosperm has 16 number of isoenzymes. LDH has 5. Alcohol deoxyhydrogenase has 4. Mechanism of enzyme action. Lock and key mechanism. Log substrate key enzyme, or it is also known as template theory, given by Emil Fischer. Enzyme will act like a key. It will act upon specific substrate, which will act as a lock. Now, enzyme substrate, okay. Now, induced fit theory, modification of lock and key mechanism, given by Koshland. The active site of enzyme is further divided into two sites: buttressing and catalytic. Buttressing supports the S substrate, and catalytic has all polar groups like COOH and NH2 and SH. Now, active enzymes can slightly change shape so that catalytic site is actually exactly opposite to the bond to be broken. Okay, nucleic acids, very important. Nucleic acids are formed by polymerization of nucleotides of nitrogen bases or not of vitamins. DNA is made up of two nucleotide strands, single polynucleotide strand of DNA. DNA is made of two now single polynucleotide strand of DNA. A single nucleotide DNA has three components, that is phosphor, then phosphate ion, then two two sugars. Okay, this forms the base of the strand of the DNA here, and then there is also a nitrogenous base. Okay, adenine and thymine. Okay, and DNA, and in the RNA, uracil and thymine and guanine. Now, ester bond is formed between the phosphate and OH group of sugar. Phosphate diester bond is found between H3PO4 and OH group of CH3 C3 of sugar of one nucleotide of OH group and C5 sugar of another nucleotide. Here you can see fifth carbon 1 2 3 4 5, again 1 2 3 4 5 5, as here there is third carbon and here there is fifth carbon. There is a formation of phosphoester bond here. See, the two strands of DNA molecule are antiparallel to one another because one strand is 5 to n 5 to 3 direction and the other strand is 3 to 5 direction, or we can say that PO4 is present at the opposite ends in the two strands of DNA. That is, if there is strand like if having this like this, then the other strand is just opposite of this, like this. Here is adenine, then here would be thymine. Just ulta kar do this O there. Okay, just ulta this now. um, and then if or we can say that PO4 is present at the opposite ends of the two strands of the DNA. At 5 end, that is of the DNA strand, always PO4 group is attached to the 3 OH group of is attached. Now, as here is 5, then here is 3. These two strands are antiparallel. 5 to 3 direction, then here rotated 3 to 5 direction. PO4 at the top, here OH at the top, OH at the bottom, PO4 at the bottom. DNA molecule has secondary structure. The double helical structure of DNA was first observed by Franklin and Wilkin. They observed a crystal of DNA under an X-ray microscope and X-ray crystallography data. Now, based upon these data, Watson and Crick were the first to make double helical model of DNA molecule out of wires and beads and got a Nobel Prize in 1953 to 1954. The two strands of their DNA molecule are quite one upon another, just like two strands of a rope in one common axis. The width of DNA molecule is 2 nanometer or 20 angstrom. The structure of a DNA molecule is just like a ladder. Backbone of the DNA molecule is made up of sugar and phosphate, whereas the horizontal ranks, that is the connection between these two strands, are made up of nitrogenous bases. What are these nitrogenous bases? These nitrogenous bases are adenosine, thymine, guanine, and cytosine. And A and T, that is adenosine and thymine, have two weak hydrogen bonds, and guanine and cytosine have three weak hydrogen bonds. Distance between one nucleotide and another is 3.4 angstrom, that is 0.34 nanometer. Now, structure of bDNA. bDNA is nothing but the common DNA. It is the most common type of DNA in cells of our body. Now, here is the structure of bDNA. There would be one turn at having 10 base pairs. Now, what is a base pair? Common simple ATGC anything is called a base pair. Okay. Now, in every between two base pairs, there will be a phosphate group on either sides. Now, that is at every 20 bases, that is P and P here, 2 at per base pair. This is whole one base pair. Okay, this is completely one base pair, and then here are two, so 20 bases are present. Now, common one very thing that every 10 there would be complete rotation of this, like this. There would be something, then it would also be here. It would just complete one rotation. Okay, it is like a ladder, circular ladder, not like how it is looking in the copy. Now, one base pair since has the length of 3.4 nanometers, so 10 base pairs, that is one turn or one pitch, will have 34 nanometer length. At every ascent of one base pair, since 10 base pair and 10 base pair complete a 360-degree rotation, so every one base pair will turn 36 degree. Now, adenine is always a pair with thymine with two weak nitrogen bonds which are easily broken down. So, in DNA molecule, whenever A T base pairs are present, these are called low melting area. Now, when the bonds are broken, it is called denaturation. Now, guanine will pair with the cytosine with three hydrogen bonds. They are not easily broken down. These areas will be called as high melting area. Chargaff's rules, very important. He made important observations on the base content and other content in a DNA molecule and gave the following rules. He said that the total number of purines in a DNA molecule is equal to total number of pyrimidines. What is purine? Purine is nothing but the adenosine, guanine, and uracil. And what is pyrimidine? Pyrimidine is nothing but thymine and cytosine. So purine is equal to pyrimidine. This may be wrong what I said. Now, if total amount of adenine is 30% in the DNA molecule, what will be the amount of thymine, guanine, and cytosine? Okay, since A at the sum of the number of adenine will be equal to thymine, so this is simply that adenine is equal to thymine, and that number of guanine will be also equal to cytosine because they form a pair and there will be no exception. And so A barabar D, to 30-30, and the rest would be G plus C, and G and C equal, so 20-20%. And this A plus G barabar T upon T plus C equals to 1. Simple thing. Now, molar amount of adenine is equal to molar amount of thymine. and so guanine equal to cytosine. Molar amount of deoxyribose sugar is equal to phosphoric acid. Ratio of A plus T upon G plus C is constant. It is used in forensic lab. In humans, this ratio is 1.55. In cow, it is 1.3. Now, types of DNA and their base pairs. BDNA, we knew that it had 10 base pair. Add ADNA, 11 base pair, and cDNA, 9 base pair. These three are RHS, and LHS, that is 2 base pairs. Okay, that is ZDNA. ZDNA is left rotating, and the other three are clockwise rotating. Now, RNA. RNA is generally single stranded. In reovirus, RNA is double stranded. In RNA, Chargas rule is not applicable because it is single stranded. Types of RNA. rRNA present in maximum amount of cell, 80% of the cell. rRNA discovered by Kunz. mRNA 3 to 5% minimum amount. Largest RNA. DNA added, then transcription, then mRNA, then translation, then protein. mRNA was first discovered by Jacob and Monod. tRNA, also known as soluble RNA, adapter molecule. Smallest RNA, 10 to 15%, it will carry the amino acid to the site of protein synthesis, discovered by Hoagland. RNA has tRNA has two structure. Now, two-dimensional was that is clover leaf-like structure given by Holle, and L-shaped structure given by Clark. Metabolism. All chemicals reactions included in the metabolism, anabolism, catabolism. We know this or nothing else. Now, a living state is non-equilibrium steady state and able to perform the work. Now, main thing important here: inhibition of enzyme action. Competitive inhibition of enzyme action. It is brought about by a substance called as competitive inhibitor, which has close structural similarity with substrate. Example: succinic acid on enzyme succinic dehydrogenase forms fumaric or fumarate. Now here the competitive inhibitor is malonic acid or malonate. Now this is a competitive here. If the concentration of malonate is more here, then it will form enzyme and malonate substrate complex, and it is brought by the competitive inhibitor. If the complete substrate will occupy the active site of the enzyme, so no formation of fumaric or fumarate would be done here. Now simply, if concentration of this is all I said about. Now enzyme succinate dehydrogenase acts upon succinic acid, which is the substrate. This is about competitive inhibition is reversible on increasing concentration of substrate again. Since it is formed, that is malonic acid has occupied the active site of the enzyme. But again, if the succinic acid for concentration is increased, then again the enzyme substrate of complex can be formed.